Since 2006, the Common Fund has established over 50 separate bioscience research programs. Each program has a dedicated data coordinating center to curate and publish its scientific results, and each has its own interface for scientists to explore this data. Common Fund programs have excelled with respect to their original mandate, which was to catalyze and transform research methods over deliberately finite funding periods. Programs have also produced immense amounts of data, which clearly has its own value apart from innovations in methodology, but using this data effectively presents a whole new set of challenges. Before the CFDE, there was no way for a researcher to even ask if there was some Common Fund program offering data sets that might be useful to their own investigations. Finding data was effectively blocked by the need to interact individually with dozens of separate programs, each with its own unique interface and research focus. And finding data sets wasn't the only hurdle. Researchers also need to decide quickly and cheaply whether or not the data sets they find are likely to be useful to their own work. Trying to understand results can quickly become intractable thanks to differences in perspective, detail, and even terminology across multiple independent programs whose approaches by design don't usually overlap in straightforward ways. The CFDE's mission has been to deverticalize Common Fund data by offering the research community a unified interface, facilitating three critical tasks: finding data, deciding quickly whether or not that data is useful, and accessing useful looking data for deeper investigation. The heart of CFDE technology is C2M2, the cross-cut metadata model, which gives Common Fund programs a universal way to describe their results. C2M2 lets the Common Fund for the first time offer a one-stop interface to explore the entirety of Common Fund research, thousands of data sets from dozens of independent programs. C2M2 is an abstract schema defining a relational database describing experimental resources. The English translation of that is that C2M2 is a standard way of describing the most important components and aspects of biomedical research datasets, so scientists with no advanced knowledge can quickly ask useful questions about the totality of Common Fund research, guided by their own research interests. Broadly speaking, C2M2 models resources, scientific concepts, and relationships. Resources include concrete things like subjects, files, biosamples, and chemical compounds. Concepts include more abstract topics like diseases, genes, and anatomy. Relationships describe connections among resources and concepts. For example, this dataset contains information about Alzheimer's disease, or this biosample had gene NEUN knocked out, or this subject was given doxorubicin. So far, 12 Common Fund programs have published C2M2 metadata through the CFDE. Here are some quick counts. For resources, we now have records describing around 5 million files, 50,000 subjects, 2 million biosamples, and 100,000 chemical compounds and substances. For concepts, we have C2M2 data describing nearly 2,000 diseases, 20,000 individual genes, which already comprises nearly the entire human genome, 65,000 proteins, 100 experiment types, and about 100 different file data types and formats. A variety of relationships connecting all of the above are also included. Because C2M2 serves as a bridge between Common Fund programs and the general research community, its purpose is really twofold, not just to make Common Fund data findable and usable, but also to make it as easy as possible for Common Fund coordinating centers to describe their data without having to expensively reorganize or reprocess what they already have. For example, different data sets linked by common scientific concepts can only be connected to one another if they're described using a common language. So C2M2 uses controlled vocabularies of scientific terms to describe key concepts. By design, we provide only one way to identify concepts like diseases, chemical compounds, anatomy, and so on. As a result, a large part of the work done by Common Fund programs to describe their data with C2M2 involves deciding how best to use these shared vocabularies. The CFDE Coordinating Center works closely with each program to ensure that their data is fairly and accurately described. Because we use universal vocabularies, programs don't have to interact with each other in advance just to ensure that their C2M2 descriptions are mutually compatible. Concept harmonization becomes automatic when everyone's using the same language, and this harmonization is ultimately what allows our users to find related data sets across the whole of the Common Fund. Virtually every part of the C2M2 model is optional and can be left blank. which lowers the barriers for programs to make their own data sets findable in the CFDE portal. They can first provide simple, basic descriptions of their resources without having to invest huge amounts of time up front creating the most detailed possible descriptions. Key resources can therefore be described and tested quickly, with more detailed C2M2 annotations added in later, over time, to better describe data and to enhance findability. Here are some links for further reading.